Chapter 19 is all about viruses. We're going to cover the structure of viruses, their reproductive cycles, and how they work as pathogens in our body. The Latin root for virus means literally poison. When we, as scientists, think about what exactly is a virus, how do we classify it, why do biologists study it, we don't classify them necessarily as living, even though they kind of fit that gray area between living and non-living organisms, and we're going to talk about why that is a little bit later on. So we would consider them to lead kind of a borrowed life because they're between living and non-living forms of matter. They don't fit our traditional definition of what a living organism is. They can only reproduce within other cells. We're going to look at what the reproductive cycle is like in a little bit. Viruses were observed indirectly way before we ever were able to get a direct observation of the virus. At the time, back in the 1800s, we could not visualize with a microscope what a virus looked like. We didn't have anything powerful enough to view them. But we started to study the tobacco mosaic virus, which was named after we figured out that it was a virus that infected it. The disease causes a stunting of the tobacco plant's growth, and then it also causes a mottled coloration of the leaves. In the late 1800s, researchers hypothesized that there was a particle smaller than bacteria that actually caused the disease. So something on a level far smaller, you can't see it with a microscope, but it's not bacteria. And it wasn't until 1935 that Wendell Stanley actually confirmed this hypothesis, that there is some particle able to infect organisms that is smaller than a bacteria. He crystallized the infectious particle. We have now named those particles viruses and then renamed the tobacco mosaic disease as the tobacco mosaic virus. One of the first experiments done, this is backtracking just a little bit, was to take extracted sap from a tobacco plant that has the tobacco mosaic disease, and they ran it through a filter, and you can see this is being run through the filter. And the filter catches larger particles such as bacteria and allows everything else to funnel through. So then they took this filtrate down at the bottom and rubbed it on a healthy tobacco plant's leaves to see if this plant in fact became infected with the tobacco mosaic disease. And what was a healthy plant became infected, and you can see this mottled coloration on where they rubbed that. And so that told the scientists that, hey, you trapped all the bacteria in your filter, so there's something smaller than bacteria that is causing this disease, this infection. So let's talk about the structure of viruses. We don't consider them cells. They don't have all the organelles that a regular cell would have. They're very different. They have a nucleic acid that's enclosed in a protein coat, and that nucleic acid can be DNA, it can be RNA, and then surrounding that's your protein coat. Sometimes they have envelope membrane on them. They can be either double or single-stranded DNA for their nucleic acid, or they can have double or single-stranded RNA. They also have the protein shell that encloses that nucleic acid is called a capsid, and it's built from capsomers, and we're going to see an image of that soon. So here's some examples of viral structure. Tobacco mosaic virus has kind of a rod, a cylindrical shape. Adenovirus has what they call an icosahedral shape. Influenza tends to be more of a round shape, and that's what causes the flu. And bacteriophages are probably my favorite. Not that you're supposed to have a favorite, but they are. And they have a head to them that encloses your DNA or your RNA, and then they have a tail to them. Some viruses have membrane envelopes that help them to infect their hosts. The viral envelopes that surround the capsid, so they're on the outside of that protein coat of the flu virus and a lot of other viruses found in animals. And these viral envelopes that the virus has surrounding their protein coats are derived from the host cell's membrane. So they have a combination of both viral and host cell membranes. And the brilliant part about this is that also in a host cell's membrane are a bunch of glycoproteins and glycolipids that have those ID tags on them that tell the cell, hey, I'm one of your own, don't attack me and don't kill me. So it's almost like a Trojan horse thing they got going on there, where they're hiding in the host's own cell membranes and the body can't detect that that cell is in fact infected. Bacteriophages are also called phages. These are viruses that specifically infect bacteria. They're some of the most complex protein coats or capsids that we find among viruses, and we will spend a lot of time talking specifically about bacteriophages as we go through this lecture. All right, so one of those tricky things about viruses is, yes, they reproduce. So they have that trait that's similar to all living organisms, yet they can only reproduce when they're in a host cell. So we consider them obligate intracellular parasites, meaning they have to be within a host cell in order to reproduce. So they're kind of parasitic in that sense, and they're within the cell. Each virus has a host range, which is a limited number of organisms that the virus can infect. That would be the main reason why when you get sick, you don't get your dog sick when you give them a hug, and vice versa. So once that virus has entered a host cell, it takes over the cell's machinery and uses the cell's own machinery to make viral proteins. So it's also going to use that host cell's enzymes, the ribosomes, their tRNAs, their amino acids, their ATPs, all their supplies, all their enzymes, and it's going to use that to make more viral nucleic acid molecules. So it's basically using the host cell to make more viruses. Those viruses can also spontaneously self-assemble new viruses. So it turns your host cell into a virus-making factory. Here's some of the brilliance of how viruses work. So the first step is your virus, which has the DNA or the RNA on the inside of that protein coat. It is going to inject its DNA into the cell, the host cell. Then the host cell is going to start to replicate the viral DNA using the host cell's enzymes to do so. So you have a bunch of copies of viral DNA now floating around in the cell. While that's going on, your enzymes are also transcribing viral DNA into mRNA, which will be used to make certain proteins that make up the capsid proteins of the virus. So it's not only replicating its DNA and making a bunch of copies of those, but it's also transcribing and translating some of those viruses' protein products, specifically that outer protein coat for the virus. Then the proteins and the DNA self-assemble into new viruses, and eventually they're going to make an exit from the cell. As I said before, bacteriophages tend to be the best understood of all of our viruses, and they're one of the easier ones for us to study. We know that bacteriophages have two reproductive mechanisms, the Lytic cycle and the Lysogenic cycle. The Lytic cycle, and remember that lytic comes from the word lysis, which means to split or to burst open, is a phage reproductive cycle that culminates in the death of the host cell by rupturing it. So eventually the end of this is going to be releasing your virus by bursting the host cell's membrane. So you kill the cell in the process and you release a bunch of these new viruses that have just been made. Phages that reproduce only by the Lytic cycle are called virulent phages. And bacteria that do have defenses against these phages, including restriction enzymes. And restriction enzymes are special enzymes that recognize this viral DNA, and they can cut up that phage DNA without damaging their own DNA. So it's kind of targeted specifically as a defense mechanism against these bacteriophages. Okay, this works the same way as that basic diagram we saw a couple slides ago. So here you have a bacteriophage, and it lands onto a host cell. In this case, it's a bacteria like an E. coli. It injects its DNA into the cell. The DNA starts to take over the cell's machinery, and it replicates multiple copies of the viral genome and takes over those enzymes that would transcribe and translate those viral proteins to make the outer protein coat. And they start to self-assemble. Once self-assembly is complete, they burst out of the bacterial cell, releasing all of these new viruses to go infect all those other bacteria cells are in close proximity. The lysogenic cycle is the other type of reproductive cycle that a virus can have, and this is where you are replicating the phage genome without destroying the host. So your viral DNA gets incorporated into the host cell's chromosome. So we would call this a prophage. So if you have your viral DNA integrated into your host DNA, that's a prophage. And then every time your cell divides through mitosis or binary fission in the case of bacteria, it will copy its own DNA along with the viral DNA. So each subsequent cell is going to have that prophage in it, or that viral DNA. Then, when you get an environmental trigger, such as stress, if an organism is stressed or in certain conditions, that can trigger that virus. It's just sitting tight, not doing anything, it's not replicating, it's not bursting the cell, it's not making proteins. It can switch over to the lytic cycle. And when that happens, you start making those viral copies of viral DNA, and you start making viral protein products and self-assembling the viruses, which are eventually going to burst out of the cell. If you have a bacteriophage that uses both the lytic and the lysogenic cycle, we would call them a temperate phage. So this is an example of a temperate bacteriophage, one that would go between the lysogenic cycle and the lytic cycle, and let's see how those two are related. So let's start with infection. Your bacteriophage injects its DNA into the host cell, and here we're seeing the DNA as a circular piece of DNA. If that piece of DNA becomes incorporated into the host cell's DNA, we're entering the lysogenic cycle. So here we see the DNA is part of the host DNA, and every time that cell reproduces, you're going to have two daughter cells, each of which are going to have that viral DNA in it. Should we get an environmental cue that causes it to stop reproducing and stop hiding out in the cell, stop going through lysogenic cycle, and to shift over to the lytic cycle, we see that prophage exits the bacterial chromosome. So it goes back to being a separate circular phage, a separate circular piece of DNA. And that piece of DNA starts to replicate itself using the host cell's machinery, and it also transcribes and translates protein products, self-assembles those viruses, and then eventually the cell will lyse, releasing all of those viruses to infect neighboring bacterial cells. In order to classify animal viruses, we do that based on their reproductive cycles. We can classify them based on whether they use DNA or RNA, and whether that DNA or RNA is then single-stranded or double-stranded. I like this table because it lays out a bunch of examples of viruses that we see, and whether they have an envelope, and whether they are considered single-stranded, double-stranded RNA, or DNA. So here we see double-stranded DNA. Adenovirus, which is a respiratory disease, does not have an envelope, whereas herpes does have an envelope. Some examples, let's see, Ebola. It has an envelope but is a single-stranded RNA, which is a template for your mRNA synthesis. So we can see there are different ways to classify viruses based on their nucleic acids and whether it's double-stranded or single-stranded. If your virus has a membrane on it, replication is slightly more complex. You have just another step. So on that surface of that membrane, you're going to have receptors. Those are going to bind with the surface of your host cell, allowing it to gain entry into the host cell. So at this point, your capsid and your viral genome are both going to enter the cell. The cell is going to be like, hey, that's not one of my own proteins. I'm going to go ahead and chop up those proteins, releasing your mRNA in this case, which acts as a template for another complementary RNA strand to be made. And from that piece of mRNA, we can both make glycoproteins, which will be on the membrane of our new virus, as well as capsid proteins, which go right inside. And we also get copies of the genome, so we make more mRNA. Again, essentially, it's the same thing that's going on in every other cycle that we've talked about, except for we're adding in that membrane component. These vesicles fuse to the membrane, which will then be used to create the new membrane for your virus, which encapsulates the capsids and the mRNA that are viral, and that releases your new virus with a membrane on it. We usually think of DNA as holding your genetic material, so it's kind of strange for us to talk about viruses that use RNA as genetic material. And we do see the broadest variety of RNA genomes is found in viruses that infect animals. We call these guys retroviruses. They use reverse transcriptase to copy RNA into DNA. So they're doing the reverse of transcription that we've learned up to this point. HIV is an example of a retrovirus, and that stands for human immunodeficiency virus, that causes AIDS, acquired immunodeficiency syndrome, which we're going to talk more in depth about. Generally speaking, the reproductive cycle of HIV works very similar to the one we saw with a membrane-bound virus a couple slides ago. So you have HIV that has an RNA as its genome, as its genetic information, and because of the membrane that's surrounding it, it can bind to the surface of a host cell, and release that virus into the host cell. Once it's in there, same thing happens as before. You have hydrolytic enzymes that break apart that protein coat, releasing the mRNA into the host cell. The RNA then uses reverse transcriptase, which is an enzyme that causes a complementary DNA strand to form based on the mRNA. So that makes DNA. The DNA gets synthesized and a complementary strand is formed. Now you have double-stranded DNA, which will incorporate itself into the host cell's genome. So it becomes what we call a provirus. Once you have that HIV double-stranded DNA, it will become part of the host cell's virus. So every time it replicates, the host cell replicates, you're replicating that DNA from HIV with it. You can use that DNA, and the virus then uses that DNA to make mRNA, which can either function to make the membrane coat, which is synthesized in the endoplasmic reticulum, or the capsid proteins that will surround the viral mRNA, or it can be used to also make copies of the RNA that would be used for the next viral generation. Within the host cell, those viruses will assemble themselves, and your membrane then forms around the virus, and it is released as a new virus that can infect yet another host cell. As we said on the previous slide, that viral DNA that gets integrated into the host cell's genome is called a provirus, and unlike a prophage, which would be in the bacteriophages, a provirus remains as a permanent resident of the host cell. It doesn't switch from a lytic to a lysogenic cycle. The host's RNA polymerase will transcribe that proviral DNA into RNA molecules, which can then function as both mRNA to make new viral proteins and as the genomes for the new virus particles that will be released from the cell.